Welcome everyone. I'm Chaitanya with GFI India. I'm thrilled to welcome you all to the All Protein Dialogues. This webinar series is co-organized by GFI APAC and GFI India. It is designed to strengthen regional collaboration and technologies that transform our global food system. We have an incredible lineup today with Dr. Smith, Dr. Hariharan, and Dr. Singh to help us navigate one of the biggest challenges in our field. Now, before we dive in, I want to share a simple observation. Walk into any grocery store in India or Asia today. A kilogram of chicken or soy chunks costs less than a plant-based meat product. That price gap is the primary barrier to all protein transition in our region, where customers are highly price conscious. And much of that gap comes from imported ingredients. We're flying ingredients halfway around the world, layering in shipping costs as well as tariffs. To solve this gap, we need a paradigm shift, and the answer is literally beneath our feet. Right here in our region, we have crops that have survived and thrived for centuries. Chickpeas, mungbeans, millets, and peanuts. These crops are already being grown by millions of farmers, but in low-value commodity cycles with no larger incentives. So, what if we could develop finished alt protein products right here? Crops grown locally, processed locally, and priced competitively. Now, it's not that easy. We're building the supply chain from the ground up. We're tackling variability, infrastructure gaps, and logistics. But the question is, how fast can we close that gap? To show us how we get there, I'm honored to introduce our speakers, Dr. Millicent Smith, Dr. Subramani Hariharan, and Dr. Gurmeet Singh. As we jump into the webinar, a quick announcements for the audience. Today's seminar will run for a total of around 90 minutes. The speakers will each present a 15-minute presentation, which will be followed by a 10-minute Q&A. On the bottom of your screen, you will see the Q&A button. Please make sure to direct your questions in the Q&A window. Also, please mention to whom your question is directed to. In the Q&A session, you also have the option to upvote questions you would like to be answered, so we can prioritize that are most relevant to the audience. Now, without further ado, let's invite our first speaker on the virtual stage. Our first speaker is Dr. Millicent Smith from the University of Queensland. She is solving the foundational problem that everything else depends on. Can we breed indigenous crop varieties like chickpea and mungbean that reliable under the heat stress we are experiencing right now? Think about it. You're a plant-based meat manufacturer. You sign a contract with a supplier. 1 year, your crops deliver 25% protein. The next year, temperatures soar well above 40, 42 degrees as we have right now. And the protein content drops to 18%. This leads problems to the supply chain, your products change, and your customers notice. That's the reason why Dr. Smith's work matters. For the last decade, she's been mapping the genetic blueprints that not only control just yield, but even protein stability under extreme conditions. She'll be discussing how resilient pulses and reliable protein and how genetic diversity can improve grain legume productivity. Dr. Smith, I thank you for joining us across the time zones. The stage is all yours.
Thank you. Thank you very much for having me. I will share my screen. Hopefully that works. Okay. As I said, thank you so much for having me today. It's a real honor to be asked to sort of speak a little bit about our work that we've been doing in grain legume crops or pulse crops. So as was mentioned, I work at the University of Queensland here in Australia, and my work is really focused on understanding how our pulse crops like mungbean and chickpea, how yield develops, so that really important parameter for our farmers, and also how that might change with different conditions and what that means for quality products as well. Just confirming you can see my screen okay?
Yep.
Excellent. I'll continue on. All right. So obviously, as has been discussed, the demand in the Asia Pacific region is very, very clear. In Australia in particular, consumers are certainly eating less meat, and the export market for our pulse protein products is growing quite quickly. And so this is something that we're all really starting to think about, is how we can start to meet this sort of surging demand. that supply side is really the challenge for me. And I'm sure many people who are listening to the talk today know much more about the challenges associated with trying to access particular pulse ingredients for these new protein products. As was mentioned before, protein content can vary, sensory properties can vary, and the supply volumes are unreliable potentially, depending on the crop. And that inconsistency is a product that I'm really interested in and that we're trying to work on as much as we can. So this is mung bean production in Australia for the past 30 years. And what you can see is that that line doesn't trend exactly like we would like, which is going up over time. It really fluctuates around 1 tonne per hectare. What we know is that the genetic potential of the crop, so if we grow the crop in a field trial, in our experimental plots, or if we use special crop models, we know that a higher yield is certainly achievable, around 2 to 2.5 tonnes per hectare. And many of our farmers across Queensland actually achieve this. The average, though, still remains around 1 tonne. And this means that if you're trying to forward plan ingredient supply, that fluctuating chart is going to be a potential challenge. The good news is that that gap that we call the exploitable yield gap is closable with both better genetics and also better agronomy, so better practices that farmers use day to day. So that's part of this gap exists because of the production challenges that farmers experience. Pests and disease are obviously really big challenges for our legume crops, but drought and heat are also big challenges that we're interested in too. I'm also interested in things like how we can ensure mechanical harvesting, which is actually becoming a big growing trend in our pulse crops across the Asia Pacific region, and obviously a big area here in Australia where we harvest all our legume crops mechanically. But coming down the line is other traits that are going to be more important. And we're seeing that this month with increasing fertilizer prices, but also increasing needs to increase the sustainability of our farm production. So trying to reduce the amount of inputs, both fertilizers, fungicides, herbicides, etc., to increase the sustainability of our farm production is going to become more important going forward. So these are all challenges on farm that we think about in our work. The challenges for future markets, we know that protein, flavor, functionality are all really important for our plant protein products. The challenge is that we don't know how these interact in mung bean and if there's any variation for us to target in crop improvement. So which bag of beautiful mung bean seed do we go to find the quality trait of interest for our next protein burger, or for our potential egg products, or for our, protein shakes? Could we find different types of mung bean that better suit these different products so we can optimize our varieties for these new markets that are coming in the future? The challenge is where we actually find those potential new traits that we might need going forward. And one place we can look for this is going back in time. As was mentioned before, mung bean, really came from the Asia Pacific region. It's been cultivated there for thousands of years. And plants have been selected for those local conditions, creating enormous amounts of natural variation. So modern plant breeding, has really just been happening in the last few hundred years, and it's focused on a really narrow slice of the diversity that's available, mostly optimizing for yield and agronomic traits. And improvement in mung bean has been quite slow compared to other crops like wheat, for example. And our current commercial varieties have been developed for high yields using very few elite lines. And so that means that there's potentially traits of interest for our food markets that might already exist in nature, and we just need to try and find them and bring them forward in our new varieties going forward. So the place we can go to look for these things are gene banks, which are extraordinary resources. There's over 43,000 different types of mung bean that are held in gene banks around the world. So wild mung bean, land races that have been grown for thousands of years, old varieties, current varieties. These things have been collected by farming communities around the world, are wild species that still haven't been tapped into yet, and they're really available for our research. And what makes that exciting from a sort of food ingredient perspective, I guess, for the people on the call, is that none of these really have been characterized for the traits that food manufacturers care about. So things like protein composition or flavor profile or protein content. And so we've tried to think about how we can characterize some of these things. One of the nice things is that in Australia and in other gene banks, we started to get DNA information about the gene bank accessions. And this really helps us to start to narrow in on some of those many thousands of accessions to start to figure out which ones might be helpful for us to look at. And what you can see here is a spinning cloud of all the different mungbean accessions that we've been working on in my team here in Australia. But then once we have those hundreds, we've got our 43,000 down to thousands and now hundreds of different types of mungbean. How do we actually find the accessions of interest? This is what I refer to as looking for a needle in a haystack. It's a really big challenge. We've got hundreds of different types of mungbean, but how do we find which ones are going to be the best for our future varieties that we could use as parents to breed those new varieties that are coming down the line? And so it's one of those things that's quite challenging. We like to do this as much as possible in the fields because this is where obviously our mungbean is produced in the end, and we get a much nicer signature about how that crop's growing and responding, and it lets us measure a big range of traits. And so the work I'm going to talk about today is from field trials that we did in Queensland on 300 diverse mungbean, which were collected from around the world. So this is the mungbean mini core collection, which was put together by the World Vegetable Centre. And as I said, we're measuring lots and lots of different things. And we do this in many different ways. The first way that I wanted to share is that we actually use drones because measuring at this scale is a really big challenge. And so we fly our UAV with sensors that capture things that even the naked eye can't see, things like how warm or cool a canopy is. And this gives us an indication of things like stress resilience. We can also get other traits like height, architecture, maturity timing. And so a single drone flight can measure hundreds of plots in minutes and give us a huge amount of data that we can use going forward for a range of different questions that we have. And so a lot of these things that I've mentioned might not seem so important to many of the people on the call because you might be interested in the end use. But I just want to remind everybody that these things are actually critical for a farmer to grow mungbean at all. Without them, a variety would have very limited grower adoption, regardless of how special it might be for its protein characteristics, for example. So things like height and maturity matter enormously for harvesting. A variety that is too tall will fall over and it won't be possible to harness that yield. And stress resilience is becoming more and more important to ensure that we have reliable, consistent yield across seasons. And so it's really important that we capture this information alongside a lot of the quality characteristics that we're interested in so we can get an understanding about how they're related to one another. In terms of some of the protein traits, we've been working with collaborators at CSIRO, which is the National Science Agency here in Australia, Professor Michelle Colgrave and Dr. Cassandra Pegg, to undertake proteomics and specifically mass spectrometry to profile the complete protein content of each accession. And so this tells us not just the total amount of protein, but the identity and the quantity of all of the individual proteins that are present within the mungbean. And that's important because different proteins behave really differently in different foods. Some might be important for texture, while others might be important for some of our flavor compounds, for example. And so by profiling hundreds of different accessions, we can then identify which ones have the signatures that are going to be most useful for specific food applications. And so we can start to hunt for these as we look for our sort of future parents for mungbean breeding. The third measurement dimension that I'm going to talk about today is flavour. So the green beanie off flavour that many people associate with pulse protein is caused by specific aromatic compounds, and the main one is hexanol in mungbean. And we've been working with Professor Heather Smythe, who's a world-leading flavour chemist here at the University of Queensland, to get some clues about our mungbeans. And so we used gas chromatography to measure the volatile organic compound of all of the different mungbean accessions. And what we saw was a massive amount of genetic variation. So this is just a little snapshot of 15 different types of mungbean. And what you can see here is the sensory threshold for hexanol, which is that red line. It's 0.005 parts per million. So above that line, some varieties or some types of mungbean are more than 40,000-fold higher than what a human can detect. But promisingly, amongst just the 15 I've shown here, there are some with much lower concentrations. And so that means that the genetics exist in our collection now to start to look for these characteristics and potentially, if we want to, reduce the amount of that off flavour that we can get in some of our mungbean proteins. So that's a really exciting finding for us, I think, going forward. So just digging deeper into the library, and I'm sharing some work here from Edward Fabrique, who's, as part of his PhD project, has been taking the seed samples from a single year in the field and looking at the variation and the genetics underpinning that. So what you can see here is 150 accessions from that mungbean mini core collection and the total content. So that's along the x-axis, and you can see the range is quite substantial. It's going from 23 to 29% dry weight. And most importantly for us, that variation is strongly genetic. So it's heritable, and that means that we can actually select for it. And Edward's done some really exciting work that shows that we can also start to build genomic prediction algorithms, which are essentially like a way of us forecasting what a potential plant might produce in terms of its protein content before we've even grown a single seed in the field. And so that's going to be a fundamental shift in the way that we can select for this trait potentially going forward, and think about the sort of way that we can start to select for protein in our pre-breeding work that we do. One of the biggest concerns when people talk about new selection targets is that you accidentally trade off something really important. And yield is obviously the thing that I mentioned before that all of our farmers care about. And what you can see here is quite encouraging data. So the genetic correlation between yield and protein content is relatively low in this selection of genotypes at this location and year. And so this is just what you can see here is some of the accessions in yellow. Some of the popular Australian varieties are labelled there. And you can see that we have some accessions that score relatively high on both yield and protein. So that's really exciting. Obviously something that we need to explore further, but that's, something that we're quite optimistic about. However, there is a trade-off between protein and starch. And so as protein goes up, starch tends to go down. And that's obviously something that's relevant for processing markets as well. I guess what I want to sort of temper this finding is that this is obviously just 1 year and one environment. And we always like to do multi-environment testing to get a better understanding of what's happening. But it's something that we want to validate further. But the early signal is relatively clear that we do see a relatively low genetic correlation between yield and protein content. In terms of sort of the proteomics, as I mentioned before, mungbean is not one homogeneous ingredient. It's a mixture of protein fractions, and each of those have really different properties. And so what the proteomics data shows is that these fractions don't all move together genetically. So this is a sort of heat map And I've blocked out some of the key information here just for proprietary reasons. But what you can see, if you keep an eye out for in the literature, it'll be out soon. But what you can see is that there's some colors that are shared and some that are quite distinct. And what this tells us is that some fractions move together and others don't. So some proteins are positively correlated, some are negative, and some are largely independent. And so we have genetic correlations between storage proteins, which vary in both direction, so positive and negative, and also magnitude. And what we're really excited about is that not all of those fractions are related to one another. And this independence is really cool because it means that we can start to potentially breed for specific protein profiles, not just more total protein. And we can potentially do that without having any trade-offs on other protein profiles. But as I said before, this is just one environment and 1 year. And so there's still a lot more work to do to try to understand this further. So I've shared some variation, obviously, that we're interested in and we want to start to think about and breed, obviously, thinking about how we bring all of these things together. So you have reliable yields, protein, the right types of protein, low off flavors, and bringing this all together in a single new variety of mung bean is genuinely hard. I'm so glad I'm not a plant breeder because it's a really big challenge. It's what we call a gene number problem. And, all of these traits that I talked about are controlled by many, many genes. And so plant breeders are practical people. They think about chromosomal blocks or inherited chunks of the chromosome. And their job is really to take all of the information about the range of traits that people like me start to pull together and start to rearrange those Lego blocks into the best possible configuration to develop new varieties. So it's a really big challenge. If we just stack 30 favorable combinations, gene variations for agronomic traits, something like yield, for example, protein traits, and sensory traits, you're looking at more than 1 billion possible ways to cross those different mung bean parents together. And that is a problem that's obviously beyond any of our imaginations. It's something that is, more than the stars in the sky. There's so many different possible ways that you could do this. And so it's something that AI is really helpful to help us with. It's really useful for us to use and try to rely on to make decisions. Because crossing mung bean is also not easy at all. And so I'm really lucky that at the University of Queensland, I've been working with people like Professor Ben Hayes and Professor Lee Hickey. They've been working on developing an AI-guided breeding approach. And as I mentioned before, this treats the genome as a set of blocks or chunks of chromosome that are inherited together. And the AI can scan all of that diversity panel, so the 300 different types of mung bean that we measured for all these different characteristics. And it can identify which parents carry the most valuable blocks for each trait category and design the crossing strategy to bring those all together before we've actually even made those crosses in the glasshouse. And so we can simulate the ideal variety in the computer, and that really allows us to bring all this information together. So what you can see here is all the different potential parents for all those three different traits that I talked about before, the agronomic in green, the protein in blue, and the sensory in red. And you can see that the AI algorithm has picked out some parents that have those different chunks of those special chromosomal blocks, those high value chromosomal blocks, and it will tell us to bring them all together into an ultimate variety. And so this variety is sort of like the sort of gold standard thing that we point towards and we want to try to achieve. We might never get there in practice, but we'll get close. And so this really helps us point us in the right direction to make sure that we're making the highest value crosses to bring together all of these different traits that are very complex genetically. So this approach we're applying in mung bean at the moment with a range of other tools and a range of other partners. to try to accelerate the work that we're doing, bringing things from the gene bank and, into improved cultivars in years rather than decades. And so I hope I just gave you a little bit of a hint about some of the ways that we can start to do that in mungbean. So this is what really all of our science is starting to build towards. Our dream is to think about varieties that are designed for our future markets, where different proteins might be used for different products, and that would deliver both reliable yield for farmers in the field and consistent ingredient quality as people, get towards the factory. And so I think hopefully I've highlighted that the genetic diversity that we need to build these varieties exists. The tools that we require also are there for us, and so the work is underway, and it's a really exciting time for us to be building this together with this big community. So just to acknowledge at the end here that this work is genuinely collaborative. I'm a mere crop physiologist, and I'm lucky to work with an amazing team of people who work in proteomics, sensory science, plant breeding and genomics, and with a great team of students and funders across the world who have really helped to contribute to this work. So if you're interested and you'd want to hear more about it, then please feel free to reach out and we can continue the conversation. Thank you.
Thank you for the talk, Dr. Smith. That was very informative and insightful. I've got a couple of questions. So, when you breed a heat-tolerant mung bean, how much variability comes from the genetics versus the environmental stress? So, for instance, can a farmer in Australia and India grow the same variety and get identical protein content, or how much of it is dependent on the environment?
Yeah. So genetics and environment is something that we're always, grappling with. It's something that we try to get an understanding of as much as we can. So whether we will ever get to a point where a farmer could grow the same thing in Australia and India and get the exact same protein content and yield, I'm not sure. But I think what we want to point towards is a variety where it's much more stable. And so we have a higher protein content, for example, and a higher yield in some environments than others. And so what we try to do is really to get a good understanding about how the genetics change across different environmental conditions and how we can start to find different accessions that might sort of help to manage that variation. So essentially that genotype A would always perform higher than genotype B in the same conditions. I think we can probably aim for that. Whether we can sort of get within exactly the same protein content within a percent, I'm not sure, but I think, one thing we can aim towards as well is sort of getting within a specification range. And I think that's something that we can certainly work towards.
And what's the typical protein content in mung beans right now? I remember seeing the slide where there was spread between 20 to 30%. So,
Yeah, that's
and
right.
how accurate is that? So, if we're going for breed that goes with 25%, um, what's the spread usually like? Is it between 24 and 26% where it's tight, or does it vary with other factors where it
I
goes
think,
from
yeah,
22 to 28 or something like that?
yeah, no, I think it's a good question. So the information I showed is for diverse accessions, but even within our commercial varieties that I showed there, the percentage sort of varies around 5%, I think, from memory, and that's within the same environment. However, obviously, agronomic management has a big role to play in the amount of protein that we can load into the grain within a crop. So things like the amount of nitrogen in the soil, the amount of symbiotic nitrogen fixation that's happening. Obviously, our legume crops are excellent at bringing atmospheric nitrogen into the plant. So there's a whole range of different nitrogen dynamics that can change the amount of protein content that's occurring. And so, as well as the environmental conditions of the day, so things like drought and heat, for example. So there's a lot of different factors. I guess the goal is to try to get the genetic variation as close as possible, so it's sort of more stable across environments. And so then you don't get such wild fluctuations in protein content.
And we've got a question from the audience. Is there a difference between what makes a good crop for traditional mung bean markets versus what the alternative protein sector needs?
Maybe in short, yes, I think. Um, I think it'd be good if we could get towards a closer, more common goal. One of the challenges is that obviously a big factor in our current markets for our legume crops is visual grain characteristics. So these are things like size, color, shape, luster. So for mung bean, how big and green and shiny it is. And those things are, are potentially important for our markets, our export markets, particularly here in Australia. But they're not necessarily important for our alternative protein products. We don't really mind what it looks like if it gets mushed up into a new product. And so I think there's potential traits that we're currently looking at that are likely to continue to be important. But in the long term, breeders starting to think about what variation might be available for new protein. It takes a long time to breed a new variety. And so there is interest in thinking about what variation might already exist for these potential new traits like protein content, and how it might be possible to bring that genetic variation into the program so there's not such big trade-offs going forward. I don't know if that answers the question clearly, but I think, yeah, there's certainly a lot of things that breeders have to look at when they're thinking about a new variety. And our traditional markets are still really driving that decision making. And in the future, I think it might change. It's just building that momentum and getting the information out there to try to change that.
We've got some other questions on the chat as well, but I'm afraid we're running short of time. So please feel free to type in the answers as we go.
Sure. Thank you very
Yeah.
much.
But thank you so much for these answers and the talk. I really appreciate you laying out the science so clearly for us. Now, let's talk about what happens next, because having the genetics and harvest is one thing, getting into the market is another, where our next speaker comes in. It's Dr. Subramani from Proyon Foods. If Dr. Smith develops the varieties, Dr. Subramani works on the processing and scales it. Proyon is doing something that sounds simple but is incredibly complex. They're taking indigenous sources like peanuts, mung beans, and underutilized crops and are developing them into protein ingredients that actually work in commercial products at scale. His perspective is invaluable. He's on the ground dealing with raw material variability, supply chain fragmentation, and he's lived the cost multipliers that nobody expects. His talk is called From Promise to Product: What It Takes to Scale Plant Proteins. Dr. Subramani, the floor is all yours.
Thank you. Thank you so lot, Chaitanya. And thanks, Dr. Millicent. I think that was a really illuminating speech and talk. I also learned some new things from you. I've been in this field for quite some time, and I've seen a lot of publications being published over the last decade, probably thousands of papers. But most of them have been relegated to just the academic vault, if I may say. How many of these papers, how much of this research is actually translated into real world applications? How much of them make revenue? What is the impact on society? So these are some of the questions that I've been trying to find answers for quite some time. And I hope in the next 15-20 minutes I can share some of the learnings that I've had here at Proyon. But before I start, while I'm sharing my screen, take a moment and just think what scale means to you. So I hope, Chaitanya, the screen is visible.
Yep, it is.
Okay, great. So thanks, everybody. I'm really excited to be here. I'm Subramani, and my talk is From Promise to Product: Scaling Plant Proteins. more specifically, I'll be talking about, I mean, we all hear about this term, plant proteins are good. They're really good. They're sustainable and a lot of things we keep on hearing. The real challenge is not discovering new sources of plant proteins. The real challenge is how reliably these proteins or proteins derived from indigenous plants, how do they behave at scale? That is the real question. We all know the market pull. I mean, this is nothing new. I mean, I just thought I'll begin my speech by saying that there's a very strong consumer shift. And we've been seeing this over the past decade consistently. I mean, for example, a lot of consumers are actively planning to shift to plant-based foods. There is a very strong market growth. The retail penetration is excellent. So clearly the pull exists. The question is, can supply chains and backend actually deliver? There is no dearth of indigenous plant proteins. I mean, it could be chickpea or it could be mung, it could be peanuts. They all seem ideal. If you look at the APAC regions, we have probably hundreds of different proteins. All of them are nutritionally adequate. They have really good functionality. I mean, functionally promising. We know that they are sustainable. They look like perfect candidates. They are abundant. Everything is there. So there is definitely a promises there. But here is where there is a small paradox. I call it the reality. If you have 100 indigenous plant sources, you can probably screen them and arrive at probably 10 or 12 potential candidates. But look at the reality. The reality as of today, my friends, is that only 3 or 4 plant protein sources have been commercialized. They are soy, which is like a legacy product. And I'll be using soy very much in my talk because I feel it is a global benchmark. Pea is there, rice and wheat is there. Which brings us to the question, what works in the lab? It often fails in real world. And this is not a minor gap. It is a systematic failure across multiple stages. So this is something that I'll try to share in the next slides. What is the promise versus reality? There is definitely a promise. But the reality, my friends, is that when you scale indigenous plant protein sources, there is definitely a big gap in functionality, in sensory. There is a big lack of standardization. We think that what works for soy or what works for pea might work for peanut or it might work for mung. My friends, we are all wrong. There is a big gap. There is a lack of standardization. And because of this lack of standardization, there is a high capital expenditure and operational expenditure when we talk about scaling indigenous plant sources. And finally, there is also brand loyalty. I mean, there's not much that we can do about it except good marketing tactics. So friends, the issue is not just science. Let me be very clear. The issue is not just about science. It is a multidimensional system problem. So let me begin. Here is where the story starts, my friends. Let me begin by asking you a question. Why does a protein that works perfectly in the lab? We have all done a lot of experiments, including myself. I have done a lot of experiments. They work fantastic. Why do they fail at scale? And if you remember, I asked you a question. What does scale mean to you? Does scale mean larger volumes? Does it mean increasing manufacturing capability? If you thought like that, yes, you are partially correct. But according to me, scaling means not only large volumes, but it also means the ability to produce, listen carefully, a protein ingredient consistently. Now, this is the word. Consistently at large volumes, stable functionality, predictable cost, and reliable supply. So it is like a holy grail of multitude of things. And all of these things must work. They must hold simultaneously for a protein product to scale, to work, to be commercially successful. So friends, it is a systems failure. So the real question that we must be asking is, why do systems fail? Now, failures do not happen at a single point. They usually happen because the system, I'm repeating once again, it is not a scientific failure, it is a systems failure. The system behaves it is a chain of interdependencies. Um, I'll tell what that means. Before that, I'll talk about 3 points. I'll cover these 3 points in the next 3 slides about why we are not able to scale up indigenous plant proteins. So the first concept I want to introduce you is something called as failure map. Remember, I told you it is a system. The system is consisting of 4 different stages. It starts from sourcing. I'll just take a highlighter. Yeah. See, you can take any plant in the world. It begins from sourcing. There can be some pre-processing, and usually it is met with extraction and purification. This is where all the process engineers and chemical engineers they come, and finally like the food technologists they come into the foray and they say, okay, fine, you have extracted, you have made, you have purified an ingredient. Let us make an application, and this could be any application. It could be like a plant-based meat or like a drinkable protein product, or it could be as simple as say protein isolate. Now, here is where I've taken the example of 4 plants: peanut, mung bean, chickpea, and millet. The breakdowns occur at different stages across the value chain, and each crop fails differently. For example, if you look at sourcing, it is very easy to procure peanut because, peanut, if you take a country like India, India is the second largest producer of peanut. And it is quite easy to procure peanut. Not so easy to procure mung bean, like as Dr. Millicent aptly put, there are a lot of different varieties. So the question a manufacturer might ask is, hey, which is the correct quality, which is the right kind of raw material I should source? So here itself, there is a failure. And this failure keeps on translating to the next stages. For example, if you go to extraction and purification, peanut is notoriously difficult raw material to work with because of its oil content. Mung bean does not have any oil, so it does not have any processing challenges. So friends, the takeaway from a failure map is that each crop behaves differently, and that is very critical. Looks good on map, but what does failure actually look like? Let us take an example. Let us take your typical indigenous plant protein. I'm assuming this is it could be chickpea or mung bean. I'll leave it up to your imagination. You can choose your favorite plant protein. I am betting that it will have some protein. It will be moderate protein, probably around 20 to 25%. It will have some starch, and there's going to be quite a bit of non-starch polysaccharides. So when you have such a raw material, it is going to absorb a lot of water. And because of this, if you put it in a extraction tank, imagine like a 10 kiloliter, huge extraction tank. You're dumping your raw material, you're putting water. This is going to this is going to increase the viscosity of the slurry. Because of that, the overall extraction would be affected. Mixability is very poor. If mixability is very poor, then you are going to have to spend a lot of energy to mix it properly. Your water usage is going to increase. What does this all mean? This means that per kg of your protein, you're essentially the cost is going to be tremendous. Since the cost is tremendous, your product ultimately fails in the market. And then we come to a conclusion, friends, this is not scalable. So the takeaway is that composition of the plant is very important, and this drives the behavior of the plant during processing. So again, I just want to reiterate that failures are not random. They are structurally linked to the composition and process interaction. Let us take the example of chickpea. In Proyon, we worked on chickpeas. Chickpea has a protein content of 22%. It is mainly composed of globulins, mainly leguminous and vitulins. And there's a fair bit of carbohydrate. I won't delve too much into the chemistry behind it. This is something we can all get on ChatGPT. But what I want to share is my real-world experience. What we found out was that because of the high percentage of carbohydrates, there was incomplete protein solubilization. There was a lot of problems with the flow behavior, which ultimately led to lower yield. And also the chickpea had very the final product was not palatable, which led to challenges. So on paper, chickpea is a perfect candidate. It is a potential candidate, but because of the high percentage of carbs and because the flavor profiles negatively influence its behavior, the product does not scale. So this is where most scale-up efforts fail. My second concept, now this is a key concept. It's called as a protein instability spectrum. What I want to say here is that I mean, like, if the three columns, they represent three different stages of scale up, from lab to pilot to full-fledged commercial. When you take a protein through all these three stages, there's going to be a lot of pressure on the protein, which would lead to loss of functionality. Now, legacy proteins like soy, they are very strong, they are resilient. But some proteins like peanut, mung, or chickpea, they are far more sensitive, my friends. And when you take them across, they can, lose their functionality. So small processing variations can lead to very large functional losses. And again, that makes scaling very challenging. The third concept is again very important. This is about mass balance, and which a lot of chemists or process engineers miss. Let's look at what's happening. Again, soybean is my favorite. It's a legacy protein. If you take 100 kgs of soybean, you do the extraction and you assume that there's going to be 80% recovery. You'll be left with around 25 kgs of protein. Now it has a byproduct which is 20 kilograms of oil. And you have around 55 kilograms of soy meal, which acts like an anchor market. You take mung, you take 100 kilograms of mung bean, and you optimize the process for 80% recovery. You have around 20 kgs of protein, which is coming from 100 kgs of mung bean. 80 kgs of fraction is still there as a byproduct. What is happening to this? It's being thrown away because there is no market for it. So if there are any researchers, PhD students, this is a potential topic for you. See what you can do with this mung bean starch. Friends, this is a very important problem in scale up. What can you do with this fraction? This is exactly the reason why some proteins like mung or peanut, they are unable to achieve price parity, because a protein like soy, we are using all the fractions and the mass balance is quite predictable. My final framework that I want to introduce is something called as the reliability equation, or systems reliability. So remember that I told about, you think about this entire process as a system, and there are 4 different stages: raw material quality, processing, functional recovery, and applications. The system's reliability, it's basically a framework to see what is the reliability of each stage. And this is a multiplicative process. It is not additive. And in scale-up, each stage must perform consistently for the system to succeed. Okay, Subramani, now you have told us what is a reliability equation, but what does it mean? So let us take an example. I'll again take the example of soybean and mung. What does this reliability equation mean? So let's say that for soybean, you have an R value of 0.95, which means that 95 out of 100 times you're going to get a soybean which meets your internal raw material specifications. For soybean, this is pretty much optimized. At all stages, the R value is 0.95. Let's look at what's happening in mung bean. The raw material is 0.85, processing is also good, functional is also good, application 0.7, on paper looks good. But when you do the multiplication, the R value for soy comes out to be 0.81. Look at what's happening to mung. It's only 0.36. This is another reason why it is very, very difficult to scale up proteins. So I hope, the previous slides gave you an idea of it is very difficult to scale up indigenous proteins. Everything's good. So what? We know how failure happens. So how do we ensure that failure does not happen? How can we prevent failure? What actually needs to work? Friends, everything needs to work. Right from being aligned on input to processing to application, everything needs to be aligned. So again, I'll talk about 3 major points here. My first point is about input and processing. So input, as Dr. Millicent also aptly said, that there's going to be a lot of variability in raw material. So as a manufacturer or as a person who is wishing to work in this field, the raw material selection, the crop selection, understanding the proximate composition, and establishing a reliable supply. This is very important. You should also give a lot of focus on processing. Remember when I told you about the instability spectrum? Design your process. We cannot eliminate instability in peanut or mung or chickpea or millet. So your processing design has to be it has to be adaptable. You have to design it uh, based on the instability of the protein. This means that you have to give a lot of impetus on the extraction process. You have to understand what is the yield. Am I getting good yield, good purity? What is the water use, energy use, and what is the robustness of the product? If these points are taken care of upstream, then what is going to be the outcome? The outcome is that you are going to have a product which is highly functional, it will be scalable, and it will, most importantly, it will achieve cost parity. So here I want to say scale is determined by early stage decisions and not downstream fixes. Friends, my second point is on establishing cost parity. And when we talk about cost parity and economics of the system economics and something called circular bioprocessing, they go hand in hand. I won't delve too much into this, but I just want to impress upon you the fact that when you consider this as a system, it is going to be a balance of yield. So let's say you have 60% of non-protein and 40% of protein. How much protein and how much non-protein are you able to extract? Can you increase this number? Can you increase the yield? So yield is going to be very important. What is going to happen to the co-products? Remember in mung, I talked about 80 kilograms being wasted. It is nothing but the mung starch. So can you valorize that starch? And finally, water and energy use. Due to lack of time, I don't think I have sufficient time to be talking about this, but just want to tell you that you're not just extracting protein, you're designing a circular system. And of course, when you talk about proteins, purity is one variable, but what customers actually want, and Dr. Millicent also talked about this, is not just protein purity, but they want a protein which is functional. So parameters like solubility, emulsification, flavor, all these parameters matter far more than just the percentage of the protein. And sadly, this is often misunderstood in early stage R&D. So I'm nearing the end of my speech, but I thought I will give you a couple of case studies of what we did right in Proyon. Reliability equation, if you remember, the current reliability was only 0.36. We took this challenge and we have optimized each stage and we have increased the reliability so that the final R value of mung is coming close to that of soy. And we had to, invest quite a bit in this. So as a researcher, you should focus on improving the reliability of each stage. Don't just focus on extraction. Proyon PP80 is our flagship protein. It is Proyon Peanut 80. This protein we completely do. It's one of its kind protein. It's made from peanuts. And the challenge was the supply chain was decentralized and the protein itself has very low functionality. Aflatoxin was the main challenge here. Again, we strengthened our supply chain. We came up with a lot of unique processing techniques. And because of the heavy investment and impetus on R&D, we came up with a very high quality peanut protein, which had a minimum of 80% purity. Most importantly, we were able to reduce the aflatoxins, and the final product was a clean, functional protein that has a multitude of applications. Okay, friends. Now you have extracted the protein, you have ensured the protein is really good. I just want to deviate a little bit. This is my second last slide. Also think about how you can use this ingredient and market this ingredient. So there are a lot of frameworks. This is, I'm entering the realm of marketing. So some things like, Ansoff Matrix, and there are a lot of frameworks. Pestle is there. Think about where your ingredient fits in. For example, like, if you have like a new market and new product, most of your products like plant-based milk, meat, they fall into this segment. So it's a high risk, but high reward. So this is my last slide. I asked you a question, like, in the beginning of my presentation. Why does a protein that works perfectly in the lab fail at scale? I hope my presentation has given you some insights. Chaitanya gave me a very difficult topic to work with, but I hope some of these things are clear. I'll quickly, go over these things. Like, you have to work on instability. Instability is a real problem. So design your models, not just for stability, not just for purity, but for stability. There are poor co-product economics. So build a full biorefinery system and prioritize sensory. So friends, I'll close my talk with 3 thoughts. Plant proteins, don't think of plant proteins as commodities. Each plant protein behaves differently. Success requires balancing taste, cost, and sustainability. And the winners in this field are those who will design integrated bioprocessing systems. So that's it. That's from my side, friends. Thank you for this, and I'm open for any questions you may have.
Thank you, Dr. Subramani. I'm incredibly impressed with how you and Proyon are navigating through these complexities. It is very inspirational for the researchers in the audience solving these challenges, and I think you gave them great ways to look forward to. I've got a couple of questions. So, when we talk about raw material variability, which is a huge constraint within a decentralized production system like India, what does that actually look like? Can you give us a concrete example on how the raw material variability affects the industry?
When we think about indigenous plant proteins, we subconsciously think about soy or pea. Now, soy or pea, they are legacy products. I want to reiterate that it is good to have a blueprint, but don't rely on soy and pea. What does raw material variability mean? It essentially means like differences in protein content, something as what Dr. Millicent also said. I'll take the example of peanut. We have been working on peanut for quite some time. And when we started this project in Proyon, we found out, like, in different regions, the protein contents were, significantly different. And it was quite difficult for us to design a robust system. Raw material variability can also mean the presence of anti-nutrient factors. For example, in peanut, certain varieties might be rich in aflatoxin. Now, this is not due to genetic or any harvesting problem. Aflatoxin is purely a post-harvest problem. It is how you store the peanut. So if the peanut raw material is properly stored under proper humidity and everything, the aflatoxins can be contained. So when you look for variability, these are, you first look at what kind of what is the proximate content of the raw material. You look for what kind of anti-nutrients are present, and there's always going to be, like, supply chain issues also. So here I just want to tell that you cannot control variability. Variability is not in our control. What we can do as process engineers is to design such a robust process that variability is taken care of. And that's exactly what we did at Proyon. I mean, like, even now we get, like, raw material which is, like, very high in aflatoxin. But the kind of processes that we employ, the kind of extraction strategies that we do in-house, it ensures that the aflatoxin is taken care of.
And how does sourcing play a role? Do you source from limited suppliers, or is it something that you said that variability is just a part of the process and we can't play much with that?
Um, Sourcing is again a big problem, Chaitanya. So basically, we had multiple challenges when we began to look at indigenous sources. The first was quantum. So we were lucky in peanut because India produces peanut in huge amounts, but it is again decentralized. So is some raw materials like soy, pea, they are quite centralized. So we had to do a lot of back and forth with vendors and identify vendors, strengthen the supply chain. We had an entire team dedicated to finding out which variety of peanut might be good, which variety of mung bean. We are also producing mung bean protein isolate, by the way. So to answer your question, supply chain is a big challenge. The only way you can work in decentralized supply chain systems is to get into the field, talk to vendors. Uh, there's not much apart from that you can do, to be honest.
And so for each of the source crops that you were talking about, peanut, mung, and other crops, what's the realistic path to cost parity with commercial soy protein isolate? Which one do you think will scale up to tens of thousands of tons actually? And what's your idea on that?
Thousands of tons. I would probably put my bet on peanut at this stage. Um, I'll give you a very generic answer here. Any raw material that has a protein content between 25 and 30%, has a moisture content of less than 10%, and has manageable NSPs and starch is a good source. So I feel there are still a lot of protein sources. I've been talking about peanut and mung and millets and chickpeas, but I still feel there are a lot of raw materials still yet to be discovered, yet to be something like fava bean. Australia is doing really good when we talk about fava bean. They have really captured on that. Uh, can we have and to the first part, like what about cost parity? Like of course, we see cost parity. You cannot expect to have a protein which would uh, kick out soy protein from the market. Realistically, that's not possible. If you want to go head on head with soy or pea, in today's world, it is very challenging. So, as a researcher or as an entrepreneur, try to find out where. So that's why I showed you the Ansoff matrix. You have to find out first of all where your protein fits. Something like mung has got a very niche market in gelation, in replacing eggs and bakery applications. Insofar as cost parity is concerned, one can achieve cost parity if they go for like a circular bioprocessing approach where all the byproducts are valorized. So if today, soy protein would be costing somewhere around 300 to 400 rupees per kilogram, and mung would be probably, I would say, 2.5 or 3x. Now, the reason why this is happening is that we are not able to capture the byproduct. Everything's going waste. So for all you young researchers and everybody out there, so take this as see whether you can do something with the mung bean starch. If you are able to valorize that, if we can come up with like a truly biorefinery based approach, yes, Chaitanya, I believe we can achieve cost parity.
We've got another question from the audience. My question is to Dr. Subramani Hariharan. Has Proyon Foods been able to make a profitable business model and fix its unit economics? And what is the monthly planned production capacity as of now?
We are an evolving company. I mean, yes, see, at the end of the day, we are still a startup. So yes, we have been having our ups and downs. We are also navigating this field. Yes, we are making revenue. We are making good revenue. And yes, I mean, the production, I mean, we have planned for expansion. And I mean, I have to consult with my production officer to ask what is the exact capacity, but we are doing pretty good, I would say.
Uh, Thank you for all those answers, Dr. Subramani. We really appreciate the on-ground perspective and the insights you gave us uh, to the researchers out here to work on the challenges that you gave us. Thank you so much for your presentation. So now we
Thanks,
have,
Chetan. Thanks,
yeah.
Chetan.
So we have the genetics and the supply chain. Now, the million-dollar question is, can we make these crops grow as finished products that rival imported ingredients? And to answer that, we have our final speaker, who is Dr. Gurmeet Singh, Professor and Dean of Research and Outreach at TDU Bangalore. He's got over 30 years of experience in R&D, including 20 years at Unilever, and he's seen what works in labs and what works at commercial scale. Uh, he knows the real question isn't just can we breed better crops or build supply chains? It's can we actually make products that taste good, are nutritious, and cost less than meat? That's texturization, the final piece of today's webinar. His talk is on blending for health and self-sufficiency, specifically how millet and soy protein isolate combinations can deliver nutrition, reduce imports, and stay price competitive. Dr. Singh, over to you.
Hi, thanks, Chaitanya. Hi, I'm dialing in from the Nilgiris. I'm here on a study tour with my class, where we go around and study the food system. It's a food systems course. We spend 2 days here with about 25 kids from food science and other disciplines. So my signal may be a little patchy. I hope all goes well. But I'll switch to a few slides and they take it from there. Yeah. Okay, so here goes. Yeah. So yeah, Chaitanya, thanks a lot for this opportunity. So I'm trying to I put some thoughts together on what does blending for health and self-sufficiency mean, right? Self-sufficiency, of course, from an India perspective, right? And specifically focusing on millets, because there's a lot of talk. What can millets do with soy? And as Subramani very rightly said, almost, I mean, you have to really look at no, soy is the lowest cost ingredient. We'll always say and all other proteins will have niche markets. Uh, Whoever goes into a niche market tries to malign soy. You'll keep hearing a lot of negativity around soy, which is not necessarily true. But it's just other people try, whether it is mung, whether it is algal, whether taking pot shots at soy, but that is the mass market protein, right? I have my university, TDU, logo there, where I do research. And then you see another logo, RIST, Rural India Supporting Trust, who are the funders who've given us very generous funds over the last 6 years or so, which has allowed us to set up probably one of the best research facilities, not just in the country, but in the world, to do research on protein products. And any of you students interested, please feel free to contact me here, whether it's for internships or whether it's for a master's program or PhD programs, or you just want to pop in, please feel. Proteins is that, well, today we are importing gluten, right? Many extent we are importing, and this, when we sit on a diverse climate resilient grains such as pulses, seeds, millets, etc. And if you look at the products today, we're focusing very narrowly. So I lost the signal sometime. Let me go back. Yeah. And so we're focusing on a very narrow, nutritionally narrow product when we look at plant-based meats, largely protein, right? And could we do something better with that? Also very ingredient heavy. We have a number of ingredients which consumers are not very familiar with, hydrocolloids, isolates, etc. And as I said, we are imported dependent. So can we rethink this whole space, right, is the question. Now, this is one way of looking at what are the dietary proteins available to us. And I have some costing here, right, as to how much would these kind of proteins cost to a consumer, right? And I'm attributing the entire cost of an ingredient to the protein, which may not necessarily be right, but just gives you a sense if you're looking at it from a protein perspective. So we have chicken here at about 500 grams, 500 rupees a gram, for 500 rupees a kg. That is a rough price for a boneless chicken. And it contains about 300 grams of proteins per kg, which works out to 1 rupee 70 paisa per gram of protein. From an egg, 1 rupee 60 paisa per gram of protein. Dairy milk, it's about 1 rupee 83 paisa per gram of protein. So those are the typical choices from animal-based products, right? And you can see all upwards of 1 rupee 50 paisa per gram of protein. Prices may differ a little bit, but that's not really the point. It's saying that it gives you a little bit of ballpark. Then you come to, say, groundnut protein, which is probably the lowest cost nut protein in India, right? And that works out to about 1 rupee 20, 1 rupee 30 paisa. We start getting into microbial proteins, algal proteins. We don't have real cost there. I've put some numbers, and maybe about 1 rupee 20, 30 paisa of the order of groundnut, probably they could start competing. If you look at pulses, right, a kg of pulse, maybe 120 rupees is what a mung will cost, contains about 240 grams of proteins in there. So about works out to 50 paisa, right? So now suddenly you're coming down. But this is whole pulse, right? This is not the protein extracted from pulse. Soy is even cheaper, right? And works out to about somewhere between 25 and 50 paisa a kg of protein. And then, as Subramani was saying, that no, if you have byproducts, so if you can sell oil of soy, which is what it does, then the leftover cake is available at 60 rupees a kg, has about 5 grams of protein, and the cost actually starts coming down to 12 paisa, right? And that's where you start entering into very interesting stuff, right? Defatted soy flour. Of course, if you are going to isolate proteins, which is what we need to do for many of the products, especially the meat analogs, then you come up to a price of about 60, 70 paisa, right? For a good quality soy isolate. Another protein which is challenging soy, could challenge soy, which has recently emerged, I don't know how long it will stay in supply chain, is a rice protein, right? Concentrate. And that comes from the bioethanol industry, because India last year used 5 billion kgs of rice, which is not a small number. We grow about 140 billion kgs, and 5 billion we give to the bioethanol industry to produce ethanol, which goes into our cars. And so the leftover of that fermentation is protein, right? And that works out to about 10, 12 pesos. So of the same cost as defatted soy flour, but a little higher protein concentration, right? So that's the protein landscape that we have available in India. I've not put the cereal proteins there yet, right? So what? And therefore, right, if you look at production of oil seeds, I mean, one interesting case I always look when you look at raw materials is, and India's self-sufficiency is oil seeds and what are we are doing with it. And this is, India produces about 40 billion kgs of seeds and beans for its oil purposes, right? About 10 billion, 10 to 12 is soy, another 10 is groundnut, another 10 is mustard. So about 30, 32 billion kgs are these three big ones, right? 10 billion, so 10 billion kgs, right? So of oil is taken from here, and about 20 billion kgs is left behind. And so we have about 30 billion kgs of oilseed cake available in India, containing about 40% containing about 40% protein in there, which works out to about 12 billion kgs of protein, which could be available at about 15 paisa a kg as a raw material, right? At 40% concentration. That works out to 8 kgs of protein per person per year that we have, that we are really not using in India. And 8 kgs is not a small number, because according to our dietary guidelines, which are about 0.8 kgs or 0.8 grams of protein per kg body weight, which means every individual should consume about 50 grams of protein per day over 365 days a year. That's like 20 kgs of protein intake is what, per person per year is the requirement, and 8 kgs is what we have just from the oil seeds, right? If we could start using them into value added products somewhere, start making isolates, concentrates, or use them as is. So that's a massive uh, uh, protein supply that we have. There are challenges to overcome. It's not that straightforward. We also need to share it with the dairy industry because they use it as a feed. But that's a very low value product. No, the farmer sells it at about 20, 30 rupees a kg. Whereas here we are talking about 300 rupees a kg by upcycling it. So that's one source. Pulses, of course, another source. Fermentation residue, I mentioned rice protein, dried spent grain, other sources. Also, grain straw, which is a waste in the agri industry, right? Converting to mushrooms is another area we are looking at. So these are all areas we are actively looking at in our research programs, right? And we partner with another few, right? And all these protein sources, right, could then be structured into an array of things, right? From baking applications to egg applications to dairy simulations, egg analogs, extruded expanded products. But the focus today probably is more of texturized products through dry and high moisture extrusion to get them look like meat. So let me start focusing there, right? But overall, this is the space. If you start looking at what are the protein sources and what concentrations they are available at, and when we start looking at low moisture extrusion, we certainly want upwards of 50, 40, 50% protein in there, going all the way to 60, 70%. And when we look at high moisture, we start at about 60, 70% and can look at isolates up to 80, 90%, right? So that's the two spaces we are interested in from the kind of raw materials that I spoke. And to get to that space, you could use two processes, a dry process and a wet process. Dry process has become popular recent times, and then wet has been traditional way of extraction of proteins. Dry basically and size difference, whereas wet separates starch and protein based on their solubility difference. And you could actually layer the two together, where you can take the dry ingredient, go from 25% protein in a pulse to 50% with using a dry extraction process, and then take it on in a wet extraction from 50 to 70 or 90, wherever you want to end up with, right? And therefore create a greener process. That's why I call this. And at TDU, we have a dry extraction process, an air classified system. We have a decanter spray dryer system for wet extraction. And then together, whatever concentration we get, we have an extruder system, twin screw extruder system to convert them into dry textured proteins or high moisture meat analogs. We also have homogenizers for dairy applications, for paneer kind of applications, for sausage kind of application, multiple equipment. But here we'll talk about, right? I'll go on to more of the textured and high moisture meat analogs, right, in my next few slides. So the initial thinking was that, hey, if we could take soy and convert it into a soy concentrate, defatted soy flour, as I said, at about 23 rupees, about 23 paisa per kg per gram of protein, one of the lowest cost proteins. If I could isolate and remove everything else, so from 50% protein in the flour, could I go to 90% protein, 95% protein? As I said, the price goes up from about 20 paisa to 60 paisa in the process if you buy from the market. Of course, there are margins built in. But if you do that and you start extruding, you end up with some material looking like, which is not very fibrous. And if you put it in the mouth, it's a little hard. It's like chewy. It lacks fixed complexity. But it does help you in getting to a 25-gram protein per 100-gram moisture serving. The remaining 75 grams, about 65, 70 grams is actually water, and the remaining 5 grams are other stuff, right? But as I said, the sensorials are not great in here. And so we start adding secondary ingredients to improve this fibrousness, to improve the texture, improve the multi-phasic system, as we call in chemical engineering and food engineering. And the most popular secondary ingredient has been gluten. And why gluten? Because gluten is, again, 70, 80%, depending on whether you have gluten or high gluten powder, you can have 60 to 90% protein in there. And so you blend the two together, you're still at a very high protein percentage, right? And you start getting some structural changes, right? But while you retain the protein content, you can still deliver 25 grams per 100 grams serving in a high moisture meat analog. But your texture is still not, maybe a little more fibrous, but still very rubbery, actually. It's moved from chewy to a rubbery texture. So springy, it comes back, it doesn't break very easily, right? And what we are looking for actually is then we start looking at, okay, if I have to replace wheat, gluten, and try and look at something else, then what should I replace with? And because the space is now open, we start looking at, hey, can I go beyond my thinking of just limiting to protein and look at other things as well? So other things include, can I reduce phytate? Because soybean has a lot of phytate. Actually, all pulses have, and beans have, legumes have a huge amount of phytate. And in the Indian context, having high amounts of phytate means that you have reduced uh, nutritional value, in the sense the micronutrients like iron, magnesium, zinc, calcium get absorbed and are not released in the food. So you have this challenge, right? So can I reduce phytates? Can I reduce raffinose family oligosaccharides, which cause cramping in the stomach, which are there in pulses, right? Can I reduce carbohydrate load, especially if I'm looking at adding millets, I'm adding pulses, right? Pulses come with a huge amount of carbohydrate. Every gram of protein has 2 to 3 grams of carbohydrates. So while we think of pulses protein rich, actually it is starch rich, right? Also, when it comes to soy, there is a big, as there are two unmet challenges in soy. One is phytates, and the other is that soy does get rancid, right? Even though it has very little oil, oil has been removed, but it leads to bitterness, and a lot of taste issues we find now in soy protein isolates are actually stemming from some rancidity in there, which we are trying to work on in our research programs. So the core hypothesis now is, can I blend soy protein with other things like millets, pulses, or other defatted oil cakes like groundnut oil cake, mustard oil cake, coconut oil cake, etc., and enable texture formation, improve nutritional density, maintain or reduce cost, and reduce import dependence, right? And nutritional density largely focus on fibers through addition, and micronutrients through a release, because we reduce, the phytates in there. But this is how, if I just focus on millet from here on, right? Because that's the title for the talk today. And you look at, right, millets, of course, have hardly any protein, right? About 9, 12, 8, so about 8 to 12% protein. This is one variety. You may have a little higher variety, but hardly any protein, right? It has dietary fiber in there. Some of it is soluble, right? And most of it is insoluble fiber, right? So low soluble, but more insoluble fiber, right? So that's what millets are. As a product for adding to meats, they have a good amino acid profile, better than other cereals, uh, but they don't bring in the protein part, right? Also, other things, people think of millets as having low glycemic index, right? Here I show glycemic index. I don't know how many of you are familiar with sugar spikes after a meal. This is my own data. I do measure I have a glucose meter on me almost all the time, and I measure and keep doing experiments on myself. So this is a breakfast meal that I consumed at 7 o'clock on 3 different days, 23rd April, 24th April, and 25th April, as you can see. And the first one, 3 parathas made of wheat. Second day and third day, meat of ragi. And you can see the area under the curve is pretty much the same. The peak height very much the same. So for me, ragi and wheat doesn't make too much of a difference in terms of its glucose spike, in spite of the fiber and other things, and the low amount of fiber that it has. Also, if you look at this curve here, is showing soluble fiber at one end and is showing phytate at the other end. And ragi, from all the ragis we have analyzed, go from about 300 milligrams per gram to about 800 milligrams per 100 grams, which is quite a high amount of, right, phytate. And you would like to be operating way, way lower, which is where rice, polished rice, and wheat and vermicelli are. So they are preferred grains because they do not bind to micronutrients. They don't make them unavailable, right? Of course, if you want high amount of soluble fiber, then you start getting into black gram territory, right? Maki dal. You must have seen, it's called, used for dal makhani. The makhan doesn't come from the makhan that is added on, but it is that the soluble fiber in it makes it look like very buttery, right? But you would need to reduce phytates quite dramatically if you wanted to make it a regular dal, right? So this gives us a space that, hey, millets actually have a lot not going for them. So why should we start using millets? The rheology that we do on millets, then these are some pasting curves that we did on on various millets: sorghum, pearl, finger millet, right? The green curve is pearl millet. The purple curve is is finger millet, ragi, right, and so on. And the red curve here is wheat. So wheat gluten has one property that doesn't dissolve in water very easily, right, at even high temperatures. So there are some challenges, and millet flours could have way better performance in terms of solubility at high temperature. Low temperature is very poor solubility. Soy protein isolate, this is the for that, has reasonable solubility even at low temperatures, right? At 20, 30 degrees centigrade. As you start hitting 60, 70, which is around here at 8-minute time interval, you get solubility of all these ramping up, right? So millet start behaving at 80 degrees, but therefore we extrude at about 120 degrees. Millet solubility is not a problem. Wheat gluten is still a challenge, right? And so millet does have this advantage of being able to create a nice melt at higher temperatures. And therefore, when you start extruding them out, we can see a big difference, right? Now you see a lot more vibration compared to the earlier pictures I had shown of soy, which didn't have that kind of vibration. And so this is sorghum from 10% incorporation to 20% to all the way to 30. 30, the product structure starts getting destroyed. Pearl millet, you have nice structure even at 30. Ragi, again, 20, 30, reasonable, nice structure that you could incorporate. We are trying now to go up to 40%. So that melt characteristic actually resulting in much finer textures than anything that at least we get with the soy alone, definitely not. And even with wheat gluten, you start getting finer, but you don't get the kind of uh, texture that we have here. And this is maybe a little complicated, but this shows the hardness, the chewiness, the resilience, the cohesiveness, the springiness, things which are in-mouth experiences. This is soy alone, very hard, very chewy, right? Very cohesive, doesn't come apart, right? And very springy like a chewing gum. With wheat gluten, this black circles, you can see the hardness has come down, the chewiness has come down, but the springiness, the rubberiness stays up there, right? Along this black circle and the square are almost coinciding. And the moment you start putting your sorghum and pearl millet, bajra and finger millet, ragi, right, you can start seeing an appreciable drop in springiness, which is very good, right? You move from very rubbery, and this drop that you see from 0.9 go to about 0.8, 0.7 is now getting you into nicer territory. Meat is certainly lower, but you still get into a nice different territory, which here. And then we said, okay, let's look at ragi a little more. It's giving us some interesting textures and interesting profiles in mouth experiences. And can we separate it into the endosperm layer and the bran layer of ragi, right? And what is interesting in these two layers is then you start separating the outer bran layer of ragi from the inner layer is look at the dietary fiber, right? The outer bran layer is now almost 25 to 30% fiber in there, right? The endosperm, hardly any fiber. And so we said, hey, what if we can start adding this stuff into our and start getting fiber also, right? And here we see, right, that this is the fiber-rich fraction we have added, and you can see the kind of vibration and the color that you are getting. And this is, of course, the starch-rich fraction that you are doing. And it creates very interesting textures. The color also changes. You can see 110-degree extrusion to 130-degree extrusion, depending on what kind of meat you want. This almost looks like red meat. I would say 110, 30% incorporation, right? Going on to darker territories, right? And so you could really create nice, interesting brown meats, which actually command a very high value, almost double, triple of normal. I mean, if you've had the black chicken, the coconut chicken, right? Uh, the price is like 4 times that of normal chicken, right? And you could get into that special territory suddenly and introduce health because you have fiber. And see, this is a curve, my glucose curve again. I do a lot of experiments. This is my standard meal, right? And this is the same meal where I've replaced wheat with fiber, right? Or added fiber, not just replaced. Same meal, I've just added fiber to it, right? 30 grams of fiber added on a 500 kilocalorie diet. And you can see how I've reduced the area under the curve and the peak size to about half, right? It's quite dramatic. And so while this was done with soy fiber, our next goal is to use the ragi fiber and try and see where we can get down to these curves. So you can see that how we can combine these things and start getting into better territory. Of course, there are a number of questions that we'll need to answer before we can actually make nice use cases of this for industry. We need a lot of, as I say, deep ingredient science, right? Processing, uh, fractionation of millets, the understanding of starch, its gelatinization, its interaction with protein network, its process sensitivity to moisture, temperature, its color chemistry, the nutrient quality. While I'm saying that by getting phytate down, we will increase the release of iron, etc., and zinc, two things which are very deficient in Indian populations, right? That's the target there. And then, of course, supply chain validation. So many of those things we are working on in our labs. And as I said, it's a call out to all of you students as well on the call. If you want to do some projects, yeah, please welcome. And especially if you want to do a master's, this is the season when you're looking for masters. I would say it's a nice area to get into for a master's program. And of course, for India, a lot of opportunity, whole muscle analogs, hybrid foods, affordable proteins. We're looking at bringing prices down of to half the price of meat in terms of protein costs, right? Actually, half the price of pulse. I mentioned pulse is at 50 paisa per gram of protein. We want to give a 25 to 30 paisa per gram of protein, right? And we feel we can get there, right, with some of the ideas that we have. And it aligns with the millet mission and livelihood mission that India has, government of India has, the nutrition security mission, the climate resilience, because things like millets and pulses grow in dry, arid areas, and therefore our dependence on crops which require a lot of water, right? We kind of diversify our food price. Last slide here, just a call out to all the students. 5 food science MSc programs verticals: the processing innovation, nutrition and health, food informatics, which is an emerging area, food conservation, another emerging area, and of course the FSSAI has launched, and Ayush Ministry have launched the food and Ayurveda platform for foods, which we also look at, right? There are internship opportunities 1 to 8 months, and of course, please, if you can dial into interested, join the India Food Systems Youth Network. It's an ongoing. We already have about 400 students from 100 universities across India who are members. We meet weekly through webinars and other sessions, interesting gamified sessions. Please join and see how it helps you into thinking, yeah, on foods and food systems. So with that, thanks a lot. Yeah, I'll go back to my first slide so that you can see my email address if you wish to get in touch with me, and happy to answer any questions.
Thank you, Dr. Gurmeet. That was a very informative session on how blends outperform the typical isolates and how this could be a potential strategy to get into.
Yeah.
I've got one question from my end. So, obviously, from the slides, we've seen that millet and soy blends work so well in the lab scale. But what challenges emerge when you scale up from a typical lab formulation to an industrial production scale? What do you envision?
Yeah. So as I said, the results we are getting because we are doing at a pilot scale and not a small lab scale, and we are recording all the readings, right? Our twin screw extruder is very extensively designed, very well designed in the sense that it captures torques, shear rates, melt pressures, melt temperatures, not at 1 point, but 6 points. So we get a lot of readings which allow us to really minimize scale up effort, right? The parameters we get are scale up parameters, in fact. So we expect that we will get similar textures, right, at scale, similar taste at scale. So we don't see scale up per se as a challenge, right? But we do see that we do need to understand this whole chemistry between, or the interaction, the physico-chemical behavior that happens in the extruder, right? Because we've done it only on a few varieties, on actually 2 varieties of millet. And here we need to understand that, hey, millet from different seasons, different geographies, etc. We really need to understand that, right? That kind of information is available for soy. It's available for wheat. It's not available for millets, right? Also the color chemistry, right? Because our extruder, while I say we understand scale-up parameters, the residence time in our extruder, it's a 1 meter long extruder. In a commercial extruder, maybe 3 to 5 meters long, right? And therefore, your residence times will increase from about 10 seconds to about 30 seconds, right? And and what will happen? Texture we understand, but what will happen to color is something. But those are not insurmountable challenges, right? Those are challenges you feel. They're science questions that you have to answer to be able to get a consistent product from here on. Right? And certainly the health benefits that I'm talking about, we do need to get to clinical studies. So we're looking for funding, right? Because clinical studies do cost like 50 lakh, a crore, etc. So let's see who will fund those studies for us, right?
Uh, Thank you so much for sharing the slides and your expertise. We really appreciate you taking the time out from Nilgiris to accommodate us. Uh,
Yeah.
but
Cool.
here's what I'm taking away from today. We've started talking about price, climate, and farmer incentives, and we asked whether it was actually possible to build a protein industry around indigenous crops. And the answer we heard from three different angles is yes, it's being built right now. It's happening in labs, in fields, in factories as we speak. A huge thank you to Dr. Millicent Smith, Dr. Subramani, and Dr. Gurmeet for joining us and sharing your expertise so generously. I also want to thank the teams behind the scenes at GFI India and GFI APAC for making this dialogue possible. And finally, thank you to the audience. Thank you for being there. Thank you for your questions and for believing in this work. We're building something together and we're so grateful for each of you. I really wish to see you all at the next All Protein Dialogues.
Yeah.
Thank you so much.
I just wanted to thank the whole team at TDU behind this, because I'm just sharing the results, but all the work is done by other faculty members, like, there is Dr. Swasti, there is Dr. Ashwini, we have postdocs, there's Anant, there's Rebecca, there's Manoj, right? So it's a big team actually of 10, 15 people who are working on this, and I'm just sharing the results. So thanks a lot to their work, right? But any questions from the audience here? One or two before. So I know we are running late a little, but is there a question? Would love to interact with the audience. I see about 20, 25 people logged on. Yeah.
There's one I have. So, when you blend these soy and millet proteins, are they chemically interacting or are they forming new complexes or cross-linking or are they just physically mixed?
Yeah, so we don't fully understand that. That's one of the, as I said, deep science areas to understand. But what we do feel as a start, what we do see is that, the various patterns that you see, the fine striations that you see, are because both of them have very different melt characteristics, right? And therefore, as you start cooling in the cooling die, they separate out into bands and into multiple bands, right? But as I showed, wheat and soy are so dramatically different that you get very broad bands. And because millets and soy are much closer in their pasting properties, and therefore you see much finer bands. I mean, that's one of our understandings. And of course, their water absorption is very different. Wheat doesn't hold water too well, whereas millets do hold water, which is why the narrower bands, right? So, and those are areas to understand the water holding patterns, and therefore looking at XRD images, SCM images, trying to understand where water is, where the starch layer is, where the fiber layer is, where the protein layer is. And that's what we'll do over the next few years. Yeah.
I really look forward to learning more on that.
Yeah.
And there's a couple of questions from the audience.
Yeah.
Do you apply any biological processes or fermentation when combining these crops?
Yeah, there are students who are working on fermentation end, primarily to reduce the phytates, right? As I said, one of the unspoken challenges of plant, so we are trying to say these are meat analogs, right? If you look at meat, why do you want to eat meat? One is protein, but you also say, hey, iron and other nutrients, right? Because iron doesn't bind to the protein or the matrix of meat. It releases it very easily, and it's heme iron, so you even absorb better. Whereas all plant proteins, unfortunately, when we start separating plant proteins from their matrix, the phytates come along with that. So soy is very high in phytates, right? The soy isolate, right? And therefore, fermentation is one way of getting them down to about half. Extrusion also cuts down a little bit, right? So we're looking at what all physical and standard interventions that we can do, such as fermentation. Fermentation adds cost, right? That's one downside of fermentation. It's another unit operation that you have to put in place, and therefore it makes it more niche. But we feel just understanding phytate reduction through extrusion processing itself, right, may help us get down, which is where we feel most industrially bought isolates are today. We are targeting about 5 milligrams of phytates per gram of protein.
Thank you so much, Dr. Gurneet, and thank you everyone for being here. You can, yep.
Sorry, I think I lost you guys for a minute right in there.
Yeah.
Yeah.
Oh, we were just thanking you for sharing your expertise
Okay.
and thanking the audience for being here. We'll
Yeah.
see you at the next session of All Protein Dialogues.
Yeah.
Thank you,
Okay.
everyone.
Thank you.
Have a
Bye.
great day. Bye.